Welcome back. Today we're going to start talking about the heart. This is our first little segment of lecture on the heart. In your book, The Heart is chapter 19. If you have not worked through the anatomy lab on the heart, I strongly suggest that you do so because I, in these lectures, assume that you're familiar with the structures of the heart and where they are, and I'm not going to take time to go back over that. So we always start our lecture with a review. So our review for today, let's look at this cadaver image of the heart. And then I'm going to ask you what part of the heart is highlighted in pink. So the components of the cardiovascular system, we've talked about blood. We're going to talk about the heart. And then the next thing is blood vessels. The cardiovascular system's job is adequate perfusion to all your tissues. And adequate perfusion means that all these tissues are getting enough blood to supply the oxygen and nutrients that they need and clear away any wastes. If we measure perfusion, we think about it as how much blood per gram of tissue per unit time. Again, if you are not familiar with chambers, valves, great vessels, and blood flow through the heart, stop here and please go back to the Heart Anatomy Lab. We also just want to give you a quick reminder about circulation. We're going to be talking about the pulmonary circulation and we're going to talk about the systemic circulation. And remember the pulmonary circulation, so blood leaves the right ventricle, goes to both lungs. Okay, that's the pulmonary arteries because they're carrying blood away from the heart. And then picks up oxygen in the lungs, comes back into the left atrium via two pulmonary veins from each lung. So that is the pulmonary circulation. And the rest is the systemic circulation. So we leave the left ventricle, go out through the aorta, go to all the tissues of the body, drop our oxygen, come back in into the right atrium. And now we're back where we started. Again, this is just my other way to look at it. Convention, we start the right side of the heart, go through the right atrium, the right AV valve, the right ventricle, pulmonic valve, pulmonary trunk and arteries, the lungs to get oxygenated, through the pulmonary veins, into the left side of the heart. So the first place this newly oxygenated blood goes, the left atrium, through the left AV valve, into the left ventricle, out the aortic valve, and into the aorta, which then branches into all the systemic arteries that are going to supply all the tissues. In the systemic circulation, this is purple because we're going to leave that oxygen, so we come out deoxygenated through systemic veins, eventually into the vena cava and back to the right side of the heart. And we start over. And again, this is pretending that you're a little blood cell. What are the structures that you pass? So in lab, you learned the location of the pericardium. This is another one of your independent tasks. So as you're studying, here's the section of your book. Learn the functions of the pericardium. You're going to also want to think about the 3 layers of the heart wall. We talked about that in lab a little bit. We are going to be focused on the muscle of the heart wall, which is the myocardium. It is the middle layer. But be sure you pay attention to the epicardium and the endocardium in your studying. So what is the heart's job? The heart's job is to pump. Okay. Specifically, it needs to generate enough force, enough pressure to move that blood around your body, around and through your vessels. It's a pump. A really nice, really efficient pump, but it's a pump. So we're going to jump straight into how do we make a heartbeat happen. Now, a heartbeat is the atria contract, the ventricles contract. That is one heartbeat. In order to have a heartbeat, we somehow have to start the heartbeat, and that happens in what we call the conducting system of the heart. So that's the sinoatrial node, etc. So in that heart's conduction system, we need to start an action potential, and it needs to spread throughout the conduction system. That's just sent the signal. Now we need to get the cardiac muscle to contract. So that action potential has got to spread from the conduction system to the heart muscle, and then we got to have a muscle contraction, which is going to be much like what you remember from skeletal muscles. Something to remember is this part happens 2 times per beat because it happens once for the atria and once for the ventricles. One of the cardinal rules of the heart is the atria and the ventricles never contract at the same time. So let's look at this conduction system. Up here, kind of right where the superior vena cava comes into the right atrium, is the sinoatrial or SA node. That is what we call the pacemaker, and we're going to see that this guy can spontaneously depolarize and start an action potential. Okay, from there, we're going to spread through the atria and then into the AV node, which has a very specific job. Through the AV bundle in the interventricular septum, and then up and around from the apex of the heart, traveling back this way. Okay. So let's walk through a little bit of let's walk through these structures in just a little more detail. So first we have the SA node, which we just looked at. The SA node is the pacemaker, which means it is going to initiate the heartbeat. It decides when. The AV node then is on the floor of the right atrium, so it's right between the right atrium and the right ventricle. The AV node is small and it transmits things very slowly. So what it's going to do is delay the spread of our action potential from the atria to the ventricles, because we do not want the atria and the ventricles to both contract at the same time. And then the AV bundle and the Purkinje fibers are really just spreading the action potential through the ventricles to all parts of the ventricles. Nothing real fancy. The heart also has what we call a fibrous skeleton, which is some dense irregular connective tissue that kind of serves as a floor right through there between the atria and the ventricles. One really important thing about this is that it is not conductive. Dense irregular connective tissue does not conduct electricity. So the only way for that action potential that started up in the atria to get to the ventricles is through that slow, small AV node. Okay. Other things this does, we can see kind of all our valves are embedded here. It can support them, and then muscle tissue attaches there. Okay. Now, if you don't remember at least the basics of the action potential from Bio 168, you need to go back and review that. I would suggest you review the one in the skeletal muscle chapters. That'll be most relevant to this, but we're not going to reteach action potentials. So I expect that you do have some basic understanding of the action potentials. So we're going to start out with the SA node. The SA node is the pacemaker of the heart. Okay. It is what we call autorhythmic, so it depolarizes without a signal from somewhere else. Okay. So when the cells of the SA node are at rest, they have a resting membrane potential. which is established and maintained just like any other cell with the resting membrane potential through the sodium-potassium pump and those leak channels, sodium or potassium leak channels. Now the cells of the SA node are kind of like a weird modified muscle cell. Okay? They have calcium pumps, which are pumping calcium out of the cell. They have got slow voltage-gated sodium channels, which were open at rest. That is not something we're used to, right? Normally in a neuron or a skeletal muscle, sodium channels don't open till the cell has gotten excited. Okay? This is the difference in this SA nodal cell. It has these slow voltage-gated sodium channels. We call them slow because they're tiny and sodium moves through them really slowly. When the cell is resting, meaning it's not having an action potential, those channels are open. Then we have voltage-gated calcium and voltage-gated potassium channels, which are closed at rest. And remember, a voltage-gated channel opens or closes really when the electrical charge around it changes. Okay. When this cell is at rest, where are our ions? And this is very similar to most of the other cells that conduct an action potential. Okay. So again, this nodal cell is not having an action potential. So the sodium is higher outside of the cell than inside of the cell. It is constantly flowing into the cell. So think about this. We've got a constant stream of positive charges into this cell. Calcium is higher outside the cell, just like a neuron. Potassium is higher inside the cell, just like pretty much every cell in the body. So this is starting. The SA node is what we call autorhythmic, which means it can reach threshold with no, what we say, outside influence. So no nerve impulse, no neurotransmitter, no signal from anything else. It can reach this threshold all by itself. And that's because those little slow voltage-gated sodium channels. Okay. So the three phases of our action potential in the SA node are going to sound pretty familiar. We got to reach threshold, depolarize, and then repolarize. So how does the SA node reach threshold? So if you remember, threshold is the membrane potential. Okay. At which an action potential will happen, or an action potential, let's say, will be initiated. So if we reach threshold, the cell is having an action potential and it can't be stopped. So when this cell is resting, those slow voltage-gated sodium channels are open. Sodium is flowing into the cell. And again, we say they're slow because they're small and skinny and sodium moves through them pretty slowly. So gradually, the inside of the cell gets more positive as all those positive sodiums move in until it reaches threshold. Now we know that when a charge changes, voltage-gated channels change. Okay. And so those sodium channels close. Again, this ability to reach threshold without that external stimulation is called autorhythmicity, or we can also use the term pacemaker potential. So our SA nodal cell just reached threshold by this positive sodium leaking in. Now it's going to depolarize, which if you'll remember, that's shooting up to a more positive charge really quickly. When we reach threshold, we open some calcium channels. These calcium channels open because the charge changed, so they are voltage gated. Calcium is going to flow down its concentration gradient and go into the cell. Positive charges going into our nodal cell means that the cell becomes more positive gets a more positive charge, and it eventually reaches 0, which is where that's action potential kind of tops out. Okay. And remember that reversal of polarity, so that going from threshold, okay, to kind of this peak of the action potential, that is depolarization. What is different about this? Okay. So we're going to take a minute and answer a little question. Think about what is different about this than neurons or skeletal muscle cells. Now, once we depolarize, then we have to repolarize or get back to resting. So once the cell reaches 0 millivolts, which is kind of its peak highest charge, those calcium channels close, and now some potassium channels open. And again, the charge has changed. So these channels open, so they must be voltage gated. It's a channel, so potassium goes down its concentration gradient out of my cell, and the cell gets back to resting. When the cell gets back to resting, those slow voltage gated sodium channels open, sodium starts leaking in, and the process begins again. Here is a graph of the action potential in our SA node. I don't know why that says AV, our SA node. Okay, so here sits our cell at resting. Here is where we have those voltage-gated sodium channels open. We reach threshold. We quickly become more positive. That's depolarization, and that's our calcium channel. So we peak out at right around 0. Our voltage-gated potassium channels open. Potassium quickly leaves the cell, okay, and we then quickly return to resting. Our slow sodium channels open again, and we start having this pacemaker potential. And the pacemaker potential is just this little area from here to here where those slow sodium channels are open, and the cell is slowly becoming more positive and getting closer to the threshold. So, interesting thing about action potentials in that SA node. If we take cells of the SA node out of the body, away from any wiring from our nervous system, they would like to fire about 100 times a minute, which means we would have a heart rate. They would like to have a heart rate of 100 resting. That is not our normal resting heart rate. Our normal resting heart rate is 70-ish. It varies from, even down in the 60s up to 75, but it's not 100. So a normal resting heart rate in a person is about 75 beats per minute. So there's something in the body that is slowing the SA node down, and that something is the parasympathetic nervous system. So even though our little SA node is autorhythmic, it can depolarize all by itself. Okay. It's getting signals from the parasympathetic nervous system that slow it down. And parasympathetic signals, remember, get to the heart through a certain nerve. And so let's take a minute and see if we remember which nerve. So hopefully you remembered that the vagus nerve carries parasympathetic signals to the heart. And the vagus nerve is always signaling the SA node to just slow down a little. And that constant stimulation is vagal tone. So again, our SA node would like to go 100 beats a minute, but it's getting constant, calm, restful pulses from the parasympathetic nervous system via the vagus nerve. Okay, and we know, we just talked about this, that these are parasympathetic signals. Sympathetic signals would increase the heart rate, and they wouldn't be going along the vagus nerve. Okay, so now we've had an action potential in our SA node. Okay. And now it's just going to spread through the conduction system. So think about this, it's just like traveling down the cell membrane. Okay. And it's just going to spread through the conduction system. So from the SA node, that little action potential is going to spread through cardiac muscle cells of the atria. Okay. So SA node fires, atria have an action potential, and if they have an action potential, that means they contract. But we spread through the atria, and we reach the atrioventricular node. The AV node, again, is small. It doesn't have a lot of ways for ions to move, so there's a little bit of a delay so that the atria can finish contracting before we start the ventricles. Okay, from the AV node, we go through the AV bundle, and then into the right and left bundle branches. And the Purkinje fibers. And from there, it's going to spread through the muscle cells of the ventricles. Okay, and then eventually the ventricles will contract, because that's what muscle cells do when they have an action potential. So this is the action potential in the SA node. This is how we start our heartbeat. And then we talked a little bit about how we're going to move that action potential through the heart's conduction system. In video 2, we're going to talk about the action potential and muscle contraction in cardiac muscle cells. It would be a good idea between now and then to have a little review of skeletal muscle contraction, because we're working basically the same way.